break it down. Remember that to this point, we've done the mouth, the esophagus, and the stomach. We've only talked about breaking things down. We have not discussed any type of absorption. So before we can absorb it and get the nutrients from our food, what form does it have to be in? If we're catabolizing it, what form does it have to be in order for us to absorb it and actually use it? Somebody said this. Monomers, the monomer form. It needs to be the smallest it can possibly be. So we're going to start with the small intestine and kind of talk about the anatomy of the small intestine, and we'll look at each of the biomolecules. So in the small intestine, this is where the rest of digestion occurs. It started in the mouth a little bit, nothing in the esophagus, proteins in the stomach, and now we're at the small intestine. The rest of the digestion will take place here, and all of the food absorption will take place here. The majority of the water absorption will take place. We've already used the term hydrolytic before, but are hydrolytic enzymes used for anabolic or catabolic processes? Are hydrolytic enzymes used for anabolic or catabolic processes? Catabolic. All we're doing right now is breaking down. All we're doing right now is breaking down. We've talked about the fact that the pancreas, we did this yesterday, the pancreas is an organ that's used both in the endocrine system, and that's where insulin comes into play. But for digestion, which is what we're using it for here, we're using it for its pancreatic juices. These pancreatic juices are going to add an alkaline solution, which we'll get to in just a moment. When we get our food all the way down to its monomer form, then it will be absorbed. When it is absorbed, it enters our blood unless it's fat. Fat does not go directly into our blood. We're going to have a different route for fats. All vitamins and minerals are absorbed as well. But they have to be small in order for them to pass through diffusion. When we look at the anatomy of the small intestine, and I'm going to go to the picture first, and then I'll come back, because I didn't realize last period I had a picture. So, here, small intestine, this open area is called the lumen. The walls of the small intestine fold. And on top of those folds are more folds. And then on top of those are microvilli. So I tried to draw it here. I did a pretty good job. You can see it's folded. Then you can see villi. And then the microvilli are the small extensions at the top. Do you recall back from cell biology what the purpose of microvilli is? Absorption. When you hear the term microvilli, we know for a fact that something is being absorbed. What if you see cilia? Movement. So a lot of people confuse those two. Cilia is movement, microvilli is absorption. So I'm going to go back. The reason for all these folds in the small intestine is to increase surface area. There's no other organ that's folded like that. The large intestine is not folded like that. The stomach is not folded like that because there's no absorption that takes place in those places. Well, except for water a little bit in the large intestine. So we have villi covered with microvilli. This increases the surface area of the small intestine by 600 times. So it allows for a whole lot of absorption to take place. Of course, by increasing the surface area, we increase the likelihood that we absorb the components of food that we need. Okay, now here it mentions that with each fold, I have my own blood supply, and then I have my own lacteal, what we'll end up calling a lymphatic vessel. Blood supply. Blood will take any carbohydrates we take in or absorb will go directly into our blood. Any amino acids that we take in and absorb will go directly to our blood. Any fats we absorb go to lymphatic vessels. We do not put fats directly into our blood. Fats will go through the lymphatics, which are always going to be coded green on your drawings and diagrams. They will go through the lymphatics and be carried back through your lymph nodes to be processed. Eventually, they will be dumped into the heart, but after they've been cleaned up. Okay? So when you're looking, and we're going to talk about each of the biomolecules, fats are never carried directly into the blood. They'll eventually get there, but not straightforward. Yesterday I touched on this, but once more, when we look at the length of the GI tract, or the gastrointestinal tract, herbivores versus carnivores, what are the differences? Herbivores have longer GI tracts, carnivores have shorter ones. Why? Why does it take so much longer to digest? Okay, yes, cellulose. These plants have a whole bunch of cellulose, and because these organisms don't have cellulase, we have to wait for the bacteria to help them break it down. Now, it also mentions on this slide that even within an individual animal, the length of their digestive tract can change throughout their life. If you're a carnivore today, strict carnivore, and then all of a sudden you say, you know what, I'm going to be a vegetarian for the rest of my life. Your small intestine, your large, they're all going to get just a little bit longer over time. It's not going to be like next week that it's longer. Okay, your stomach's going to be upset just a little bit, but over time it will increase in length because it's going to need more time to digest those foods for you. And vice versa. If you go from being strictly vegetarian to then a strict carnivore, your digestive tract will shrink slowly, but eventually over time. Okay? Now, we did stomach, small intestine. These two right here are accessory structures. Pancreas is going to release pancreatic enzymes. It's also called pancreatic juices. The main component I want you to pay attention to is bicarbonate. Bicarbonate is alkaline. Is alkaline acidic or basic? Basic. Alkaline is basic. So the pancreas gives us basic. The liver will create bile. The gallbladder stores the bile. So two different things. Liver makes it. Gallbladder stores it. But bile is also alkaline. Why do I want the pancreas and the gallbladder to contribute alkaline substances? To neutralize the acidic ones, the ones that are coming straight from where? The stomach. And what do we call that substance that comes from the stomach? It has hydrochloric acid in it, but what do we call it altogether? Chyme. Chyme. When chyme makes its way into the small intestines, it comes in just little spurts, and immediately we need to neutralize it. Why do we need to neutralize this chyme? It's coming in at like a pH of 2. Why? It will literally eat through your intestines. So it's protected while it's in the stomach, but as soon as it makes it into the small intestine, it's acidic enough to eat through it. So we need to neutralize it quick. So it mentions that the bicarbonate is used to neutralize the acidic stomach chyme, which is what we just said. It also mentions that bile is used to emulsify fats. That's a weird word because we've been saying digest, digest, digest. Fats start off big. When we add bile, it just breaks them down into smaller fat droplets. Okay? We want fat droplets to be super, super small so that they can then be absorbed into the lacteals. So bile is responsible for breaking down fats. Bile alone. All right? And this, I still kind of want the one with the liver. But the liver would lay right here. Gallbladder about here. There's one duct coming from the gallbladder and one coming from the pancreas. Right here, the chyme would enter. This is the first portion of the small intestine. We call this the duodenum or duodenum. Either one is acceptable. When the chyme is in here, immediately the gallbladder flexes, the pancreas does as well, and they mix in that highly alkaline, well, it's not just a solution, the bicarbonate ions along with other digestive enzymes to neutralize this immediately so that it doesn't cause any damage. Those will also help break down further whatever components are responsible for breaking down, like bile and the fats and the pancreatic juices and carbohydrates. Okay, we're going to talk about 3 of the 4 biomolecules. Basically, we've already said this, but we're putting it into words, and I'm giving you just a little bit more detail. First biomolecule is carbohydrates. Carbohydrate is fancy for sugar. When we take in really big sugars, if they came from plants, we call them, and if we take in really big sugars that came from animals, we call them what? It's a really complex sugar that comes from a plant. A starch. It's a really complex sugar that comes from an animal. Glycogen, yeah, they're both right there. First bullet. Complex starches, plants. Starch, anything is plant-based. Glycogen, anything is animal-based. Can you immediately use that information? Those biomolecules? No, they're too big. So we have to break them down. Where does digestion start for carbohydrates? It starts in the mouth, and it continues in the small intestine, and then it is absorbed in the small intestine. So mouth and small intestine for carbohydrates. Mouth and small intestine for carbohydrates. What form does that really complex starch or glycogen need to be in before you can absorb it? Monomer form. And a monomer form for a sugar is a monosaccharide. Okay? Is everyone okay with those terms? Alright, this is showing you that here I have my monosaccharides. So here's my intestines. I've broken it down into these monosaccharides, and that they're going from a high concentration to a low concentration. So they're being absorbed into the blood. What type of diffusion is this? Why is it facilitated and not just simple? It's using proteins to get through. So this is facilitated diffusion. Does it require ATP or not? No ATP because it's going from high to low. It's going from high to low. So that's carbohydrates. Starts in the mouth, ends in the intestines, and is absorbed in the intestine. Small intestine specifically. Okay? Proteins. Protein digestion begins in the stomach. We talked about that yesterday. There's pepsinogen, which is highly acidic. Acids will denature proteins. And denature means to do what? Change its shape. By changing its shape, I change its function. Right. Okay? It mentions that the pancreas will secrete additional digestive enzymes, trypsin and chymotrypsin. What does chymo kind of sound like? Chyme. Okay? We're mixing that there. Again, those are going to help to break down further proteins. What is the unit that a protein needs to be in before we can absorb it? Amino acid. The amino acid is the monomer for protein. And they will be absorbed just like the carbohydrates through the process of facilitated diffusion. So for proteins, it's stomach and small intestine. Digestion is in stomach and small intestine, and absorption is in small intestine. So here, I have my small intestine. You can tell these are amino acids because it's clearly labeled amino acid, and it's going from a high concentration to a low concentration, and it's going through a protein, so that makes it facilitated diffusion. Yes. Okay, this is the drawing or diagram that I wanted. Right here would be your stomach, dropping just a little bit of chyme in here. The very first segment of the small intestine is called the duodenum or duodenum. Here, the liver creates bile or synthesizes bile, and it is stored here in the gallbladder. As soon as the duodenum gets the signal that chyme is on its way, the gallbladder will squeeze, it'll contract, and bile will make its way directly here to mix with that very acidic chyme. The pancreas will also be stimulated to release pancreatic juices in here along with that chyme to neutralize it as well. So this is the very first part. It's neutralized, and then we begin that further digestion. Someone asked this third period. I want to make sure everyone understands. Small intestine is broken into three portions. The duodenum is the very first part. The jejunum is the middle, and the ileum is the very end. It's a long organ, but still it has three parts. So when you were looking in the lab at small intestine, it could have been any of those three would have been in the small intestine. Jejunum, duodenum, or ileum. Last one, lipids. When I taught you lipids, I said that they're going to be triglycerides. And we talked about how triglycerides were formed and how they were broken down. Digestion of lipids is entirely in the small intestine, using bile for emulsification, but then it also mentions pancreatic lipase. How do you know this is an enzyme? ACE. And of course, as Eman said, this is an enzyme that breaks down fat. So, nonetheless, we're taking these large fats and we're trying to break them down or emulsify them into smaller versions. They will aggregate into chylomicrons, small little fat globs, and then those will be taken in by the lacteals. Exocytosis, active, transporter, passive. Exocytosis, active or passive? Active. Active. So it takes a little bit of work to get these in, or get this out into those lacteals, and the lacteals is the formal name for the lymphatics found only in the small intestine. Lacteals are the formal name for the lymphatic vessels found in the small intestine. Lacteals are the formal name for the lymphatic vessels found in the small intestine. Yes, for fats. Right. Yes, ma'am. So here you can see that it doesn't look like the other diagrams that we had. So we have these fats that are being further broken down, emulsified, and then through the process of exocytosis, they're leaving, and they're entering not red, they're entering green. So this is a lymphatic vessel. So again, fats do not go directly to your blood. They will eventually make their way there, but they do not go there first. Okay, last slide. Right after the small intestine comes the large intestine. The large intestine also has three regions. The cecum, which is the very beginning. The colon, which in us horseshoes around our small intestine. And your pig, it's not that same layout. Your pig has a bunch of small intestine and it has a large intestine off to the side. Okay, so it doesn't always look the same way, but there still has the three regions. The cecum, the colon, ascending, transverse, and descending for us. And then it goes into the anus. Well, it'll go sigmoid, rectum, but anus is the opening to the toilet. Ooh. I said the three regions, but I forgot to tell you what it does. The large intestine is responsible for resorbing water. That's the only thing that is absorbed there. In the event that you've eaten foods that are plantish, like vegetables and fruits, that you couldn't digest, the bacteria have an opportunity to continue to kind of break those down for you. Good thing about bacteria is a lot of times they produce vitamins that are good for you. Bad thing about bacteria is they also produce flatus, and flatus is the fancy way of saying extra gas that causes you to fart. Yes, fart. Okay? So those large intestines, that's what it's mentioning there. All right? This is where I needed us to get today. Does anyone have any questions? The appendix is attached to the cecum, which is the first portion of the large intestine. Additional questions?